welcome to another episode of the Omics cast. My name is Alberto Leda, and today we're going to talk about graph genomes. As usual, if you want to follow me on Twitter, my Twitter handle, twitter.com/Alberto. If you want to contact me on LinkedIn, I'm happy to answer you any questions you have. And this slide deck is available upon request. We're going to talk about graph genomes and how this graph genomes field has started and evolved in the last few years. A lot of this happened due to the 1,000 Genomes project, which was the first large-scale project to collect variant information across human populations. Now, this project, which took place already a few years ago, over a period of a few years, generated a very large resource of insertions and deletions mapped to human populations. And so different individuals have different insertions or deletions, different places in the human genome. If you take the human genome as a linear reference, which is, if you want, the summary of all of us. These large research created the opportunity to describe this linear human genome sequence reference as a populations-based, graph-based object, meaning there are different paths to go along the different chromosomes if you add up all this information we have from the different populations we've sequenced so far. And then this instigated the development of software to construct and query these graph-based genome references. And then there's the opportunity to evaluate these graph-based approaches against the linear approaches and see what are the pros and cons. So I'm going to do a quick intro about this, which is based mostly on Eric Garrison's PhD dissertation, which is available online. And he's got a figure that shows a genome that diverges through generations. And basically, there's variation accumulating in different generations. And so if the reference genome is linear, then if you add more individuals to this individual reference, you can draw it as different variation graphs and with different paths for the different individuals here. And so one way of representing this, and this is one image in that PhD dissertation, is how different individuals have different paths to go from one bit to another bit of the genome. And here they show one where all these individuals shown here in different colors have these two stretches of DNA in common. And then there's one individual that goes and jumps straight into this stretch number 6 here, which is this one here, goes from here to here and then directly to here. And the other individuals have different combinations of this. So for example, this one has 1, 2, 3, 4, 5, and 6. The other ones, most of them, well, all of them miss number 4, this bit of DNA here, etc. And so this is the visualization for a haplotype in the HLA gene. And this is a different way of looking at the different individuals in different colors and how some of them have or don't have some of these stretches in the graph. A slightly different one here for a longer stretch of DNA and how in different colors you have the different individuals having different paths to go along the region of the chromosome. Now, if we construct these graph-based approaches and then we map short Illumina or BGI-MGI reads into it, which method does better? Which method has a better job at mapping these reads correctly? For example, aligners like bwa-mem, they use either in pair-end mode or in single-end mode a linear reference of the human genome. Do they do better than methods to align reads to a graph-based approach? Well, this is an initial assessment of that. And here they show the SK1 yeast strain. And they show how having the true positive rate and the false positive rate here, wanting to be perfect at this very top left corner and here, how these graph-based approaches here, cactus for the SK1 or cactus drop, which is different parameters for cactus, does better than the linear approaches, in this case, blue for bwa-mem here, right? So that's an initial one. Now, I'm going to show you the results of a more recent evaluation here done by a group in Oslo, in Norway. And what they show, they convey that the more you add information to these graphs, they can explode combinatorially, but that there are ways around this, and then they do an assessment of 3 prominent graph-based read mappers, and they compare them with the linear read mapping approaches. Now, for the graph-based approach, they compare 2 tools: the VG tool developed by Eric Garrison during his PhD at the Sanger Institute in Cambridge in the UK under Richard Durbin, and another approach, similar approach, called here 7 Bridges, done by the company 7 Bridges Genomics. And they looked at it in comparison to a linear reference-based method, bwa-mem. And then a third method for mapping histograph-based reference is HiSat2, which uses hierarchical graph for genome and gene index, and they also compare this to the others. To do this, they simulate reads, and they simulate reads with variance and without variance, and you'll see how the plots compare when they do that. There's details in the paper on how they did the simulations. There's a couple different ways in which they did it. Main conclusion is that VG from Eric Garrison outperformed 7 Bridges and HiSat2 on previously proposed benchmarks, and will show the rates in this next few slides. And then bwa-mem by default is tuned for speed, but they show how tuning bwa-mem for MapQ scores obtained by running Minimap2, a more recent version of mapping software, works for long and short reads. They show that it performs about as well as VG. Okay, so 2 different types of simulations done by the VG sim and by MITI. And shown in blue is the VG results, shown in pinkish purple 7 Bridge results, and shown in green the HiSat results. So wanting to be at this top left corner to be perfect, 2 positives and false positives, VG does substantially better than the 7 Bridges approach here. And then HiSat doesn't do too bad here, appallingly bad if the reads are simulated by VG. How about linear mapping? Well, if this is the VG result, and we compare it with bwa-mem untuned here, bwa-mem untuned, it will be fast. It will do the read mapping quite fast for you, but it'll underperform compared to VG. But if we tune the linear mapper to adjust for sensitivity with the tune parameters using Minimap2 to adjust MapQ scores, then it's about the same as VG. As you see here, the 2 colors overlapping for most of the stretch, right? And here it shows by splitting into all reads with variance, reads without variance. We can see here quite close when we compare VG and the linear mapper tuned to do better in these graphs. So the linear mapper, the reads without variance, which you could allege is simpler than with variance, does slightly better, but marginally better than VG. Whereas when you compare it to the reads with variance, then the linear mapper does worse, right? And VG comes on top here. Who are the people involved in this? Eric Garrison, left to right here, did his PhD with Richard Durbin at the Sanger Institute in Cambridge in the UK. Benedict Patton is the author of Cactus and other tools that were mentioned in this and other papers. The guy in the middle did his PhD with Ewan Burney at the European Bioinformatics Institute in Cambridge in the UK. And I believe this is Adam Novak, and I'm sure someone will correct me if he's not. Also, some of the, as you can see, 3 of the people involved in developing tools for graph-based representation of genomes. So as you can see, this is something that's ongoing, and with the promise of improving the data, the results that we get from sequencing genomes and mapping them to see the differences to other individuals, and we'll keep an eye on it to see how this field evolves. Follow me on Twitter, twitter.com/AlbertoVileja, or ask me any questions you want on LinkedIn. You can contact me on LinkedIn via this URL. I'm happy to answer any questions you have. And a reminder that this slide deck is available upon request. Until next time, goodbye, and hope to see you in the next episode of the Omics cast.